My name is Rachel Creighton, and I'm also a student at the University of Washington. I work in a lab focused on mucosal immunology and biomaterials. So what that means is I'm designing a new way to deliver an HIV vaccine in the mouth. It's really cool, and if you want to hear more about it, you can come to my talk on April 26th. As a graduate student, I get really specialized training in how to do a very specific type of research. The Engage program gave me another type of training that graduate students rarely get. And that is learning how to talk about my science in a way that's engaging, enlightening, and informative. You can learn more about our innovative program at engage-science.com. And now to our second guest. We have reached a point in human history where precisely modifying our DNA is no longer theory or science fiction, but a reality. Clinical trials are underway using modifications of patient DNA to combat diseases such as HIV and certain cancers. And scientists are beginning to see the emerging potential for this technology to be used on the unborn to prevent untreatable genetic diseases. This reality of gene editing has produced a range of emotional responses, from excitement to fear. But many of the helpful applications of this technology, from cheaper and more effective scientific research to enhance food production, are still largely unknown to the public. Our next speaker will discuss ways that this controversial field is working towards goals that aren't so controversial and may improve the lives of millions. Please give a warm welcome to Warren Anderson.
Hi, everyone. Excuse me, I just got to. Okay. So. Oh no, I'm gonna get it. No, this is a good opener. Okay. All right. So, thank you very much for the wonderful introduction. So, I work on gene editing technologies to study immunology. And I'm going to tell you a little bit about what that means. So gene editing is a technology that's really changing the world as we know it in ways that a lot of people aren't aware of. And I want to bring you guys into the process of how that works right now and show you how I use it to study the immune system and some other ways that it's being used currently. So what is gene editing? Well, it's involved in a lot of different processes that some people may have heard of, like gene therapies or genetic modification with plants. But one of the best responses I've gotten yet when asked what someone thinks of when I say gene editing is Jurassic Park. Yeah, kind of. I mean, it's a little bit more advanced than what we can do yet. But anybody else have any ideas? What comes to mind when I say gene editing? Splice. Gattaca. That's a good one. Anybody else?
Photoshop.
I like all of these. All right, so Gattaca, that's a common one that people tend to think of. On the money. Yeah, some people think about manipulating human DNA and even designer babies, where what the future of humanity is born in a test tube and we choose what traits we like. This is obviously controversial and something that needs to be considered, but I use gene editing in a way that I don't think is quite so controversial. I work in the School of Medicine, and when I see gene editing, I see medicine. Now, medicine's great. I don't think there's anybody here that hasn't used it at some time. I'd say that's a safe bet. But what's better than medicine? In my opinion, it would be immunity. And medicine really helps our own immune systems do the heavy lifting. It might give them a boost, but they're the ones that are going to have to go in and clear out a pathogen. Now, a pathogen depicted here in yellow can be really anything from bacteria to virus to parasite, anything that makes us sick. And the visual you've got here is of the immune system trying to attack that and clear it out. But what does our immune system look like? That's a big question. There's a lot of pieces, more than I can talk about today. But we can go over a couple of the main players. So here we've got an image of chess. And I tend to see our immune system constantly playing a game of chess against the environment. keeping with this, we see a little bit of a medieval battlefield. And if we think of our immune system like that, we can see our knights in shining armor, which we will call our T cells. So these are the guys that if you are infected with a virus or a bacteria, they're going to go in and just naturally clear that out. They're going to do the heavy lifting themselves, and they're going to go get right in and dirty. Then there's the B cells. Now you can think of these guys as sort of the archers, and they're going to work at a distance. They do a lot of things, but one of the things that they make is these molecules called antibodies. And they're going to float around your blood and look for anything that they want to stick to. And this is usually something that will make you sick. So if they see if there's a bacteria in your blood, B cells will usually make antibodies that go and stick to it and keep it from growing or help the immune system get rid of it. So obviously, you got your little medieval warriors. They need a castle, right? Well, our castle in this case is going to be our bones. And these guys all originate from our bone marrow. So inside all of our bones are special cells called hematopoietic stem cells. And these can grow up to become anything. They can be any part of our blood. They can be red blood cells. They can be platelets. They can be T cells. They can be B cells. These, like any other cell in our body, has 23 sets of chromosomes that make up all of our DNA. This codes for every protein that makes up a human, and it makes us us. That encodes 20,000 different genes, 20 to 30,000, and that makes about 100,000 proteins. Each gene can make one or more proteins. Some of these proteins aren't so important. If we lost them, we would never notice. We'd be perfectly healthy anyways. But some of them are absolutely critical. And so you may be wondering at this moment, how do gene editing, DNA, and the immune system all come together? And to tell you that, I'm going to tell you a story. There once was a little girl that was born. And on the surface, she looked perfectly normal. She had all 10 fingers and toes. She was taken home by loving parents. And then something happened. She started getting sick a lot. And doctors looked, and deep inside her DNA, she had normal-looking chromosomes, all 23 of them, normal size. But in one of these chromosomes, there was a gene. The gene had a very important function. It made a protein that made the immune system form. And this gene for this girl was missing a small piece. Without this protein, she had no T cells, no B cells. So normally, if she was infected with a virus or a bacteria, the immune system would show up and fight it off. But without these, the pathogens could grow and grow and grow and grow until she got very sick. So doctors told her parents that the only thing that they could do for her was to put her in a bubble, cut off from the outside world. She was diagnosed with a disease called severe combined immunodeficiency, or SCID as we commonly refer to it in my lab. This disease is pretty horrible, and there is no cure other than a bone marrow transplant if you are lucky enough to have a match. But one day, a scientist came along and offered to help. The scientist took a sample of the little girl's bone marrow, took it back to their lab. And into a little cocktail, the scientists mixed two proteins that act just like scissors, that allow them to cut DNA wherever they choose, and a copy of the DNA that the girl was missing. Scientists took these components and inserted them into the hematopoietic stem cells in her bone marrow. And inside each cell, the proteins went to work, and they cut out the damaged DNA. This was replaced by the healthy DNA that the little girl was missing, and the genes started working again. It made the protein that she didn't have. These cells were then taken and put back into our little girl, and her immune system formed. And she didn't need her bubble anymore. She was cured. So this is Evangelina. 5 years ago, she was brought to the UCLA Medical Center, struggling to get through her first year of life. She was diagnosed with SCID early on in her life, and her parents were lucky enough to enroll her in a clinical trial using a therapy almost exactly like what I described. It worked. So this is her 18 months ago. Without this intervention, she wouldn't have survived at this point. The drug that she took, or really the therapy, is now so commonly used, or used well enough, that it's called Strimvelis, and it's available in some European medical markets. On different trials, it saved another 29 out of 29 babies that have been treated with it that have the same type of skin that she had. So, this is a wonderful story. This is something that myself as a father, I live for. This is life-changing. But one of the situations that this makes me think of is SCID is extremely rare. So in the US, there's about 1 case of SCID out of every 58,000 live births. And this is actually a combination of 13 different forms of SCID that makes this statistic. If we were to treat every single one of them, we need 13 different types of cures. So this is exceedingly rare. I was wondering, when I started my research, is there a way that we can take this technology and use it to help a lot more people? So there's another genetic type of disease that affects 1 in 20 in the US. That's autoimmunity. So we talked before about the immune system being kind of like a medieval battlefield. And sometimes in these fights, if you look at old pictures, they can be really dusty, really chaotic, and really confusing. And if you're in a bit of a fight like this, it might be really kind of easy to accidentally kill your teammate, your fellow soldier, instead of the person that you're trying to get. And in autoimmune diseases, this is largely what happens. So there's a lot of autoimmune diseases. There's over 80 different kinds. Anybody here know anybody with an autoimmune disease? Yeah, I think they've affected us all in one way or another, whether it's something that us or a family member struggles with, or it's somebody that we know. It's affecting people that are in the media, such as Selena Gomez and Seal, who struggle with lupus, and Sonia Sotomayor and Jay Cutler, both live with type 1 diabetes. And the way these diseases affect the body can be quite different. In type 1 diabetes, the pancreas is confused by our T cells as an invading pathogen, and they move in and destroy all the cells that make insulin. And so people with this disease now have to have insulin injections throughout their life to be healthy. Whereas in lupus, we are stuck. There we go. All right. In lupus, our B cells might make antibodies that bind all over our body to proteins that naturally occur. And this can cause a lot of problems itself. It can damage our kidneys. It can cause blood vessel obstructions. And then even worse, T cells can see these and go and attack all over. And this can produce a number of different pathologies, including a red rash that's very common to see in people that are having a lupus flare. So what causes autoimmunity? It's a great question. There's a lot of thoughts on different contributing factors for autoimmune diseases. But the more we learn, the more complicated the picture gets. We do know one thing, though. Certain genetics are involved. So if you have a first-degree relative, you are much more likely to get an autoimmune disease. It doesn't mean you will get it. A first-degree relative that has an autoimmune disease, you are much more likely to get it. Doesn't mean that you will get it for certain. But if your sister and your brother has it, then your odds are greatly increased. And so we need to know why. And we can study this by taking groups of people that have autoimmune diseases, we'll call them our patients, and groups of people that are very similar to them in age and ethnicity and where they live in society, and call them our non-patients because they don't have the disease we're studying. We'll take DNA samples from everybody and sequence them all, and we'll find lots of little hotspots around the genome that are different in our patients, but not found in our non-patients. These are sites that we would say are genetic risks, because they seem to track with making you more likely to have one of these diseases. And then we'll take these genetic risks one at a time and start to study them. And so the way that we do this, and I've actually done this in other laboratory positions, is we assemble groups of people that don't have this risk variant, and then we find people around that do have this risk variant. We take blood samples from them, and then we isolate out their T cells, cells that I personally study. And we ask a number of questions about what makes these genetic risk people more likely to get this disease. Is it that their T cells are more eager to fight? They walk around with these big swords and they just gotta swing. Do they have poor visibility? Can they not tell their host from a bacteria or a virus? But one of the problems with this approach is we don't have that many people with some of these genetic risks. They could be very informative and tell us a lot about what's happening in these diseases, but it's very hard to find them. At the Benaroya Research Institute over on First Hill, they study autoimmune diseases specifically, and they have 2,000 plus different donors at any time ready to give blood samples. And for some of the risk variants that we want to study, we might have 3 or 4 people that we can ask for blood from. So this is very difficult to put together. And as a result, we know a lot less about the genetic risk than we do about normal healthy tissues. So this is where my research comes in, and a way that I'm using gene editing technology to try and solve this problem, where I just have a person that wants to give me blood to study. And from that, I can isolate out their T cells. And then look at them on a genetic basis and give them the same treatment that Evangelina got. Now I can change which gene I'm targeting and manipulating, but essentially, I'm able to change their DNA and produce 2 types of cells from the same person. One that doesn't code for any risk, genetic risk, and one that does. And then I can look at these under a microscope and ask the same questions that we would have before. So we may ask, how big of a deal is this actually? There's no smoking gun. There's a lot of these different genetic variants around. There's dozens really that are of big importance to me. But here's a good example from a gene that I am studying right now. It makes a protein called PTPN22. And on this screen, it just looks like a blob. I don't know what this blob really is supposed to mean. There are people out there that do. But I do know that one small difference in this protein that is based on the DNA can make this section of the blob extend out and be a little bit longer and a little bit thinner. And because of that, people that carry this genetic risk variant have double the risk for type 1 diabetes. But interestingly enough, in the same gene, if they have another risk over here, or sorry, not a risk, another variant, that makes this little line that's in the middle of the box a little bit flatter, then they have half the risk for lupus. So this protein is telling me that there's something important going on here that we need to study, but we don't have people that we can call on to provide us samples to do it. And I think this is a big deal, because not only is this gene involved in these 2 types of diseases, it's also involved in primary vitiligo, which affects the skin. It's involved in rheumatoid arthritis, affecting the joints and the soft tissues. It's connected to Graves' disease, affecting the thyroid. Myasthenia gravis, affecting the nervous system. And even something like primary Sjogren's syndrome, which is very common but often not talked about, which affects the tear ducts and salivary glands. And this is only about half the diseases that this one gene is connected to. So this is millions of people that are impacted by variants like this that are trying to tell us something about their diseases. So my distinct hope with this type of research is that one day we can put together information on these genetic traits so that we can walk into a doctor's office and the doctor can look at your DNA and put together a risk assessment for an autoimmune disease. And then they can monitor you for the rest of your life, saying you have a 10% chance of an autoimmune disease versus a 0.01% chance. We can know to be on the lookout for risky symptoms and treat with current therapies that are cheap and effective as long as we get them early enough. Or maybe we'll get lucky. We can take all these diseases that we were talking about before. Maybe we'll be able to find a common pathway that they all need to use before a disease can develop. And we can come up with a drug that blocks all of them and use that same genetic information to fight dozens of diseases all at the same time. But next time somebody brings up gene editing, I hope that you'll think about this methodology and just see this as one of the ways that it's being used to hopefully change modern medicine and change the way that we try and improve the health of millions of people. So with that, I want to thank everybody in my lab, my funding from the NIH and the Benaroya Research Institute. And of course, it is technical books, the Engage program, and CS Demo. Thank you. I'm happy to take any questions.
How do you edit the genes? Is that with CRISPR or?
Yes, the technology that I use is mostly CRISPR. That's probably the word that in the news that you'll hear frequently, but that's actually one of about half a dozen different options right now, but it's the most user-friendly, so you're going to see a lot of that. Yes?
Can you talk a little bit about what the gene editing process actually is, like for a patient? What does that therapy look like?
So for the case of SCID that I described, that's actually a gamma retroviral vector. So you would basically take a virus, take away all the parts that lets it replicate, and just use it as an empty bag to deliver the genes that you have. And in the case of that type of therapy, there's no actual gene editing with CRISPR involved yet, but there are some of those going on in clinical trials at the moment. Does that answer your question?
Yeah, partially. I guess, like, how is it delivered to the patient?
Okay. Usually it's done with a sample that's actually biopsied or drawn from the patient, like I showed here, taken to a separate lab. And there's some companies that are trying to set up shops in different hospitals across the nation where they'll do that in-house and then inject it back into the patient. Thank you very much.